So we're going to continue looking at the urinary system here as it relates to the anatomy of the kidney, looking at the functional units within the kidney and then the urinary bladder itself. So when we look at the functional unit of the kidney, we have to start with the renal pyramids. And the renal pyramid is going to be the area in between the renal columns. In this area, we're going to have hundreds of thousands of nephrons. The nephron itself is going to be comprised of multiple parts to it. But we are going to have within the pyramidal structure is multiple nephrons collecting together in what's referred to as the collecting tubules, and then exiting in what's referred to as the collecting duct. The collecting duct will junction together through the papillary ducts in the renal papillae to form the minor calyx. And so every minor calyx that you see within the kidney is going to come from a single pyramid. Multiple minor calyxes are going to junction together into the major calyx, and each lobe of the kidney is going to have its own major calyx. So we're going to go further down into the nephron itself here. In the nephron, we're going to have two principal structural areas. The two principal structural areas are referred to as the renal corpuscle and the renal tubules. The renal corpuscle is going to be made up of the glomerulus and glomerular capsule, plus all of the surrounding tubular networks. What we have to remember here is that the corpuscle itself is not the glomerulus. So the corpuscle is not going to be doing the filtrations. It's the glomerulus that's going to be doing the filtrations. The renal tubules is any of the ductal network that is contained within the nephron. So let's take a look at the glomerulus itself. So the glomerulus itself is going to be a cluster of capillaries coming away from the afferent arteriole, heading to the efferent arteriole, that's going to be housed within the glomerular capsule or Bowman's capsule. In this glomerulus, we're going to have a distinct structure of basal membrane. The distinct structure of basal membrane is going to be a combination of sinusoidal and fenestrated capillaries. So we're going to have both the small little sinusoidal holes as well as the fenestrated slits within the capillaries. That's going to allow for movement of materials out of the capillaries into Bowman's capsule or the glomerular capsule itself. The glomerular capsule is an impermeable, simple squammal structure. We need it to be impermeable so that any fluids that move into the glomerulus will head into the proximal convoluted tubule and not back out into the juxtaglomerular tissue. If we're to go ahead and dive into the actual histology of the glomerulus and of the corpuscle region, note that what we have is we have the glomerulus structure, the capillaries, surrounded by Bowman's capsule or the glomerular capsule, with all of the tubular network surrounding it. The tubular network will also have a host of capillaries associated with the tubes, the paratubular capillaries. This tubular network and capillary network develops a osmotic condition around the capsule and around the corpuscle so as to draw fluids into the nephron to undergo the process of transformation, allowing for the development of urine through the process of urination. If we were to go back to the picture and take a look at the glomerulus itself, the glomerulus itself, in terms of the capillary network, is made up of fenestrated and sinusoidal capillaries that's going to have the fenestrated basal membrane, so it's going to have these big, huge clefts within it that's going to allow for anything that is smaller than a blood cell to be able to pass into the lamina that is held together by what's referred to as the podocytes. The podocytes form a visceral layer of connective tissue. In between each of the podocytes, there are extensions of the cell itself, which form a filtration slit around the fenestrated basal membrane. The extensions of the podocytes we refer to as the pedicels. The pedicels overlap each other in such a way that they narrow out the distance in between the pedicels and the fenestrated capillary networks, thereby diminishing the size of materials that can go ahead and exit the capillaries and enter into the glomerulus. And so if we were to take a look at this histologically, now what we've done is we've jumped up in magnification. So we're actually looking at an electron microscope picture of the glomerulus here. And so within the glomerulus, we're going to have the capillary. That capillary is going to be surrounded by the filtration membranes of the podocytes with their pedestals. Note the space in between and surrounding the pedestals. The only way for stuff to get into Bowman's capsular space, into the capsule itself, is to pass through the slit membrane and in between the pedestals. And so if you notice, the erythrocyte stuck within the capillary is far too large to get into and through the pedestals, which means that we should not see blood and blood cells, in particular erythrocytes, heading into the filtrate. And if it doesn't get into the filtrate, it should not be found within the urine. So that's what's going on at the glomerulus. But let's take a look at the nephron itself. So the nephron is a cluster of tubular structures that is made up of simple cuboidal epithelial cells that have microvilli on them, just like what we saw with the cuboidal cells in the liver. They're there in order to increase the total surface area so as to increase the rate at which materials are able to be exchanged. We have distinct areas of this tubular structure that's going to perform distinct functions as relates to the overall function of the kidney. Stepping away from the glomerular capsule, from Bowman's capsule, is a structure referred to as the proximal convoluted tubule, abbreviated the PCT. This proximal convoluted tubule turns inward medullally, form is referred to as the loop of Henle or the nephronic loop. The loop of Henle has two distinct limbs to it, and the two distinct limbs are going to dictate the overall function as it relates to a process known as the countercurrent multiplication system that leads to the concentration and dilution of urine based off of hydration status, in which we have the descending limb, which is going to head towards the medulla, and then the ascending limb, which is going to head away from the medulla. The descending limb is highly permeable, whereas the ascending limb is impermeable. The only way for materials to exit on the ascending limb is through active transportation, and is usually simply electrolytes heading out on the ascending side of the loop of Henle. The loop of Henle will then lead into another set of convoluted tubules. This time here is the distal convoluted tubule, abbreviated the DCT. The distal convoluted tubule will lead to the collecting tubule, and that collecting tubule will lead to the collecting duct. Now, that's the tubular network, but that's not just that's just not part of, that's part of the overall function of what's going on here. We also have this highly vascularized region. So it's the same as what we saw with the alveolus. It's the same as what we saw with this lobule within the liver. We have this high vascular network surrounding the cells that are going to be involved with the excretion. This high vascularization of the network leads to removal and reabsorption of fluids and materials from the nephron. That leads to the concentrating of urine through the process of urination. Within the loop of Henle, we get a secondary mechanism that comes into play, which is referred to as the countercurrent multiplication. The countercurrent occurs because the blood flow is occurring in the opposite direction to the flow of urine within the filtrate, where we're going to be able to exchange materials until we get to a isoosmotic condition relative to the tissues that surround it. And so what's happening is that we have an ever increasing osmotic condition as we head towards the medulla, and then we have an ever decreasing osmotic condition as we head towards the capsule. This change of concentrations is going to dictate the movement of fluids out of the vasculature into the nephron, and movement of materials from the nephron back into the vasculature, so that we have the correct concentrations of ions and water based off of the needs for the body. Now, in terms of overall function, a majority of everything that goes into the filtrate is reabsorbed within the proximal convoluted tubule at the initiation of the urination process. So we've formed the urine, and urine is then going to exit through the calyxes, through the pelvis, down the ureter, and it's going to be held within the urinary bladder until it's time for micturition. The urinary bladder itself is going to be a organ very similar in structure to what we saw in the stomach, which means we're going to have a multiple muscular layer. We're going to have an area that's going to be rugate, that's going to allow for expansion of the bladder without disruption of the integrity of the bladder itself. We're going to have, just like what we saw with the alimentary canal, we're going to have layered tissues. So we're going to have a mucosal layer, we're going to have a submucosal layer, we're going to have a muscularis layer, and then we're going to have a serosus layer, sometimes referred to as the adventia layer. The ureters themselves are going to enter on the inferior posterior edge of the urinary bladder in what's referred to as the ureteral openings. The ureteral openings will allow urine to flow through the trigonal regions and then laterally and superiorly fill the bladder. As we fill the bladder, we start to put pressure onto the baroreceptors within the sphincter regions. The sphincter itself is a dual sphincter that is partially separated by part of the urethra. The urethra is going to be the tube exiting away from the urinary bladder. The neckal part of the urinary bladder is where we have what's referred to as the internal urethral sphincter. The external urethral sphincter is housed within what's referred to as the urogenital diaphragm. This urogenital diaphragm is going to have a slight anatomical difference in terms of dimorphic difference between males and females. The trigonal region of the urinary bladder is going to be secreting an alkalinic mucus because urine is highly acidic. If we were to look at the muscular layer, we have what's referred to as the detrusor muscles. The detrusor muscles are going to be smooth muscles that are going to contract in such a way so as to collapse the urinary bladder. They function off of a stretch reflex. The urinary bladder itself will expand and can expand to between 700 and 1,000 milliliters. However, it most often will expand to much less than that, usually a couple hundred milliliters before micturition is going to be initiated. As we exit the urinary bladder itself, coming away from the urinary bladder, we get the urethra. The urethra is going to be sexually dimorphic, which means that male urethras are going to be different than female urethras. The external sphincter for the female urethra is much closer to the urethral orifice as opposed to the male urethral orifice. The elongation of the urethra takes place during fetal development, during sexual dimorphic development due to spikes of testosterone. The urethra itself is an impermeable but highly elastic tissue. So while the ureters are not very elastic and we have to worry about kidney stone development within the ureter blocking urine flow into the urinary bladder, the urethra itself is a little bit more elastic and will allow for those calyxes to pass. The way in which urine is released through the urethra is due to a combination of sympathetic and parasympathetic signals involved in a reflexive behavior known as the micturition reflex. The micturition reflex is a partially cognitively controlled reflex in that we are constantly sending signals to that external sphincter that says, do not relax. We get a combination of sympathetic and parasympathetic stimuli that will innervate the detrusor muscles as well as the urethral sphincters. And then we have the somatic nerve from the pudendal nerve that's going to innervate the actual external urethral sphincter itself. The internal sphincter is more regulated based off of pressure than off of cognitive or autonomic control, whereas the external sphincter is regulated by strictly external control. You learn to regulate the relaxation of the sphincter starting around the second year of life, with mastery somewhere in the fourth or fifth year. And then, just as with other reflexes, because the nervous system starts to degrade, it starts to drop. There's a combination of both sympathetic and parasympathetic regulation on this pathway where you can cognitively override the necessity to micturate as long as the reflexive stretch response has not reached the external sphincter. Once the stretch reflex has hit the external sphincter, it's very hard to regulate micturition. Once micturition has initiated, unless cognitively altered, it takes about 20 seconds to void the bladder through micturition. That has to do with the fact that the diameter of the urethra will be proportional to the anatomical volume of the bladder for the individual. And they've actually done some really cool studies, and by they I mean the zoologists have done some really cool studies where they've looked at voiding times for mammals across various mammal species, and they found that regardless of the size of the mammal, it's 20 seconds for voiding to take place. And that has to do with that proportionality between the urethra diameter and the volume of the urinary bladder itself.